What if one of the most overlooked tissues in the human body actually contains valuable regenerative cells? Your wisdom teeth, often removed and discarded, contain living mesenchymal stem cells with remarkable regenerative potential. These cells are known as dental pulp stem cells, DPSCs. They reside within the pulp tissue at the center of teeth and belong to the broader category of mesenchymal stem cells, or MSCs. Like MSCs found in bone marrow or adipose tissue, they possess the ability to self-renew and differentiate into multiple cell types. Wisdom teeth are particularly rich sources of these cells because they are typically less exposed to environmental stress and wear compared with other teeth. When extracted under sterile conditions, the pulp tissue often still contains viable stem cells capable of proliferation and culture. From a biological standpoint, mesenchymal stem cells function as multipotent progenitor cells. Under the right biochemical signals, they can differentiate into osteoblasts, chondrocytes, adipocytes, and even neuron-like cells. Mechanistically, these cells respond to molecular signaling pathways, such as WNT-V-catenin, TGF-V, and BMP signaling, which regulate their differentiation and tissue regeneration potential. Within dental pulp, these pathways are involved in repairing dentin and maintaining tooth vitality, but in laboratory settings, they can be directed toward bone regeneration, cartilage repair, or neural tissue support. In addition to differentiation potential, mesenchymal stem cells from dental pulp exhibit paracrine activity. They release growth factors and cytokines, such as VEGF, FGF, and transforming growth factors, which can stimulate angiogenesis, modulate inflammation, and promote tissue repair in surrounding cells. This secretory function is now considered one of the key therapeutic mechanisms in regenerative medicine. Current research has explored the use of dental pulp stem cells in areas such as bone regeneration, dental tissue engineering, nerve repair, and even potential applications in neurodegenerative disease models. For these reasons, some medical and dental centers now offer tooth stem cell banking, where extracted wisdom teeth are cryopreserved so that their stem cells may potentially be used in future regenerative therapies. Of course, wisdom teeth can still cause clinical problems such as impaction, infection, or crowding, and extraction is often medically justified. But from a biological perspective, these teeth are not merely evolutionary leftovers. They can represent a biological reservoir of regenerative cells. As regenerative medicine continues to evolve, tissues once considered disposable may become valuable sources of therapeutic cells. If you enjoy deep scientific insights like this, subscribe to the channel and stay connected with Science for Fun Academy. And if you want exclusive study materials, quizzes, and advanced content not available elsewhere, consider supporting us on Patreon. Your support helps us continue delivering independent, evidence-based science education. Thanks, and see you in our next video.